All right, so this is going to be the next episode in the evolution series, and in this one, we're going to be touching on some of the works of Elliot Sober. And essentially, what we're going to be doing here is we're going to be exploring a puzzling kind of aspect of inferring common ancestry. And sometimes inferring common ancestry seems to conflict with real biological data. We'll explain that through the use of ribosomes, and in future episodes, we'll touch back on this point. Also, this episode is going to draw a lot on the previous episodes, specifically episode 5. So just to be aware that you have to have awareness of those episodes to continue here. So, inferring common ancestry, right? Now, scientists often ask a key question. How can we logically tell if different species come from a common ancestor? Essentially, what are the assumptions that underlie this kind of inference? A lot of the works by Elliot Sober touch on this point, right? The logic behind evolutionary biology, essentially. And so, in this particular presentation, we'll examine Sober's findings and touch on a counterintuitive paradox that Sober kind of brings up and he highlights. And then, kind of, we'll see some real examples, like, again, from ribozymes that illustrate his view. In biology, it's common to infer that certain major features or events happen only once in life's history. We say that they happen only once because they're so rare that they can't happen multiple times, right? And so the first example is Luca, and Luca stands for last universal common ancestor. And we say that Luca arose only once, and that's because all life shares core features, right? So genetic codes, ribosomes, etc. And that implies that, life originated once and all known organisms descended from that origin. A second example is mitochondria. It's widely believed that the endosymbiotic event forming mitochondria occurred only once about 1.5 to 2 billion years ago. All mitochondria is, within diverse organisms traced back to one particular origination event. So what this is basically saying is that like the origination probabilities of Luca and mitochondria are extremely low, particularly because they're so rare and complex that they're absolutely needed for life. It's inconceivable to suggest they independently originated multiple times. Professor Martin, he actually says rarity is a desirable property of endosymbiosis because mitochondria arose only 1 in 4 billion years, so only once in 4 billion years, roughly the same rate at which life and the solar system arose. So what this quote shows is that mitochondria, as well as the origin of life, are extremely rare on the order of 1 in a million years of magnitude. In his view, this rarity is expected. If something as complex as mitochondria happened more than once, it would be surprising. And so we assume that such complexity doesn't really repeat, which is why we infer a single origin event. Now, when we infer common ancestry, we must distinguish between what we observe and what we hypothesize. This is quite an important point. Observed similarity by itself doesn't tell us anything about how something arose. When we look at similarities in base pairs in DNA sequences, when we look at similarities in anatomical features, when you look at certain fossil records and similarities found, in certain species, we're not looking at their causal history. We're inferring their causal history, right? We're asking, did it come from one lineage splitting? In this case, that would be common ancestry, or from two separate lineages hitting on the same solution, right? So that's independent causes. The thing is, we need a framework to decipher and choose the best explanation for the observed data. It's not simple enough to just find similarity and, just jump straight to universal common descent. For more data on that, you can just go on the last episode where we talk on origination probabilities. So consider this thought experiment from the next couple of slides. Imagine there are two researchers, Nancy and Sam, and each discover a new gene in a different species. So Nancy finds gene A, species X. On the other hand, Sam finds gene B, another species Y. Now, these genes perform a very similar function and are quite vital for the organisms. Now, Nancy and Sam don't know each other, so they don't share their findings at first. Nancy makes a hypothesis. She says that gene she found, gene A, arose via a extremely unlikely evolutionary pathway in her species X. Essentially, she's saying that gene A is a one-time event, like a miracle that happened only once in 4 billion years within that lineage of species X. Now, Sam, on the other hand, he's unaware of gene A and he's working with gene B. And so he makes his own hypothesis and he makes the same exact hypothesis. He says that gene B arose via a similarly unlikely one-time pathway, the lineage of species Y, right? So both Nancy and Sam believe that their genes are unique and really rare in their lineages. Each hypothesis involves an extremely improbable origination. At this stage, they have no reason to suspect a connection between gene A and gene B. However, eventually, Nancy and Sam learn about each other's findings and compare the DNA sequences of gene A and gene B by doing a sequence alignment. They discover that gene A and gene B are actually very similar by all standard criteria within biology textbooks and amongst biologists. This strong similarity means that the genes are homologous, right? And again, homologous here meaning that they are likely derived by the same ancestral gene in the past. Now, this creates somewhat of a conflict because Sam made a hypothesis that his gene, in other words, gene B, was extremely low origination probability. He says it was a 1 in a million probability event, right? Likewise, Nancy said the same thing about her gene. So the conflict is that they both can't be true. If A and B are essentially the same gene and it's inherited by two species, then there was really only one original evolutionary event for that gene, and not two independent events. So either species lineage or species Y's lineage had the original gene, and the other got it through common ancestry. But again, Nancy and Sam can't both be right. So then the question is, whose origin story is actually right? Did the gene first evolve, Nancy's hypothesis, under species X, or is Sam right? Is his hypothesis right? And did it evolve under species Y's lineage, right? Now, to resolve this, let's assume they bring in an expert called Professor John to help them figure out whose hypothesis is correct. Professor John examines gene A and gene B and applies a basic principle that's often applied in evolutionary inference. That principle states that if two structures are very complex and have high levels of similarity or are nearly identical, then it's effectively impossible that they evolved twice independently. This is the same principle I brought up in the last episode. So if you guys are forgetting, go ahead and watch that once again. The point here is that if something is extremely complex and pretty much identical, then it's impossible that they evolved twice independently. Dr. Patterson says convergence between molecular sequences is too improbable to occur, just as similarity between sequences is too improbable to be explained except by common ancestry. This is the argument from complexity. If two structures are complex enough and similar in detail, probability dictates that they must be homologous rather than convergent. Based on this logic, Professor John concludes that gene A and gene B share a common ancestor. There was only one origination event for this particular gene. So at least one of the original hypotheses, either Nancy or Sam, must be wrong, right? Because both genes didn't arise independently. Now, this results in a paradoxical situation, right? Nancy and Sam each have a plausible story that their gene arose once in their own lineage. But after comparing genes, we're sure that there was only one origin in total, not two independent ones, right? So far, we can't tell which lineage had the original occurrence. Was it gene A or was it gene again, gene A and gene B are essentially the same. So knowing that they come from a common ancestor cannot reveal whether the ancestor was in species X's line or species Y's line, right? And so there's no evidence favoring Nancy's scenario over Sam's. Both are equally likely given the data. So where is the paradox? Professor, he explains the paradox like this. He says that if you notice closely, Nancy and Sam each argued that the origin of the gene was extremely improbable, right? Now, it was precisely this assumption that, such an origin is, like once ever miracle kind of thing, that made the argument for a single common origin so strong that similarity was found, right? So, in a sense, the more unique and implausible they each consider their gene's origin, the more powerful the evidence for a shared origin becomes once we saw the genes were exactly the same. So essentially, what it's saying is that the less knowledge we have, the stronger our case is for common ancestry. Why is this paradoxical? So normally we think of having more knowledge, right? Like when we have more knowledge, we think that that kind of helps us make stronger inferences. But it's actually the opposite here. Our inference becomes stronger when we lack particular knowledge. We can't conceive of a way for two independent things to have occurred simultaneously, given that they're rare. We conclude that they must have had a single origin event. That's the core of Sober's paradox. Inference of common ancestry can become stronger when the trait in question is thought to be very hard to evolve. It's the less we understand about how a complex trait could originate, the more we believe that the two organisms sharing that trait must be related. And so you can kind of formalize this, into a statement. This is actually found in Elliot Sober's book, Evidence and Evolution, where he states the more improbable it is that an ancestor has trait T, the stronger the evidence that X and Y's having T provides for their common ancestry. So what he's saying here is that if we think of an ancestral organism, right? If we think that an ancestral organism couldn't easily have a particular trait T, and again, it's some trait that is very unlikely to evolve from scratch, whether that be mitochondria or whatever it may be, then seeing two modern organisms, let's say X and Y, both having that trait is a big clue that they inherited from a common ancestor. The harder it is to evolve independently, the more we favor the single origin via common ancestry. And so, again, this captures what we see with Nancy and Sam's gene experiment. The gene was presumed to be extremely unlikely to evolve even once, but the fact that both species have it forces, the conclusion that it evolved only once and was shared. Now, to say this in a more simple way, the less we know about the origin of that trait, the more miraculous or unlikely we believe it to be, the more confident we have that the two organisms sharing that trait got it from a common ancestor. This is exactly the paradox. Our lack of knowledge leads to more certainty of common ancestry. I have this slide here because I think it was a very important with regards to this topic. Professor Van Valen says something very interesting. He says, we don't observe common ancestry, we observe similarity. Homology is a hypothesis, not a direct observation. Our reasoning depends on assumptions like T is unlikely to evolve twice. And so, again, that's the whole point here. It's like we're already assuming that the origination probability is very low. That's the first thing. And we're also assuming that things that are low means that they're impossible, right? And that's the key issues with homology as an inference. Now, going back to paradox, we can pinpoint a few more things. The probability of trait T arising independently in two separate lineages, again, it's extremely low, right? That's the assumption. Now, in our scenario, both Nancy and Sam assumed an origination probability that was extremely tiny. It was this tiny probability that makes the two independent origins seem almost impossible. If you don't understand that, go back to the previous episode. I also used the same product rule in the previous episode to expand on it. If the probability that gene A could originate in species X by chance is P, and the probability for gene B in species Y is also P, then the probability that both happened independently is P times P, which is P squared. And so when P is very small, then p squared is even smaller. For example, if each is 1 in a million, then both happening independently is 1 times 10 to the power of 12, which is virtually zero. The common ancestry hypothesis only needs the origin to happen once rather than happen twice. For common ancestry to be true, only one improbable event needs to happen. One improbable event is way more probable than two improbable events happening separately. So statistically, common ancestry wins out given those specific probabilities. Now, there's another interesting point to note here is that essentially the fact that one of the researchers he says the fact that probability of a multiple evolution of independent complex structures is low does not mean that complex organisms should not evolve at all. He notes that we often keep these origin probabilities close to zero, which raises the question, how did they even happen in the first place? If you're allowing miracles in one sense, why do we just stop there? And also, if we're going to allow miracles in the first place, then what's stopping the theory from being unfalsifiable? What's stopping the theory from being acceptable to any new light of data? That's the whole issue with miracles. If we're already invoking a low origination probability and saying it's a miracle, that implies we're already accepting miracles to begin with. On simpler terms, we can't lean too hard on this whole probability scale because we're already accepting a low probability to even begin experimentation. I thought this was an interesting point, which is why I included it here. Let's move on to a particular case study. By having a case study, we can pinpoint some of the flaws in this kind of thinking. In this case study, we're going to be utilizing ribosomes. And in future episodes, I'm going to be talking a lot more on ribosomes and the new evidence that has come to light with regards to ribosomes. But for right now, this is going to be a simple overview. What are ribosomes? Ribosomes are complex machines all life uses to make proteins. Every organism uses ribosomes for protein translation, and this trait is quite universal. No alternative mechanism has been found to do the exact same job as ribosomes, right? Even in most ancient lineages, the ribosome is quite preserved, right? In terms of not only its structure, but even its role. The ribosome is so complex that the idea of it evolving from scratch is already daunting. Now, the idea of it happening independently multiple times in two separate origins makes it even more unlikely, right? Pretty much unthinkable. So, the universality of ribosomes is considered one of the strongest pieces of evidence that all life shares a common origin, right? Because all life has ribosomes, it's very unlikely that these complex structures called ribosomes can, yeah, evolve multiple times independently. So it must have all evolved from one common ancestor. To quote Professor Jonathan Einstein, he says there's essentially no way that two independent origin of life events would both end up with the exact same complex ribosome. So he says there's no way that two independent origins of life events would both end up with the exact same complex ribosomes. If life started twice independently, it's astronomically unlikely that both lineages would converge on using ribosomes for translation. Therefore, the fact that every organism has ribosomes means that the only possibility is a single tree. So, again, what he's saying here is that in terms of our paradox, let's say trait T was ribosome, right? And let's say that probability of T arising independently is essentially zero. What he's saying is that all organisms have ribosomes. That is evidence E. According to our reasoning, E strongly favors the one common ancestor hypothesis over any hypotheses of multiple independent origins of life. Now, this is exactly how most biologists would also interpret it. The universality of the genetic code and ribosomes points to a single origin for all known life. We are very confident precisely because an alternative scenario would require an unbelievable coincidence, like two separate evolutionary miracles producing the same ribosomes. However, that's actually not the case, right? The reason why I chose ribosomes is because there's a lot of new data coming out on ribosomes, and they're showing the opposite, right? There is a particular kind of ribozyme called a hammerhead ribozyme, which is a small RNA molecule that can catalyze a reaction. It's like a self-cleaving RNA enzyme. So it's much smaller than a ribosome, but it's very complex, and it's made of RNA. So the hammerhead ribozyme was first found in certain plant viruses. What's surprising was that scientists found similar hammerhead RNA sequences in various different organisms. They found it in worms, crickets, newts. These organisms are very far apart on the tree of life. So the occurrences are very sporadic. Like not all organisms, but this ribozyme hammerhead structure seems to be popping up in very few disparate lineages. This raises a big question, right? Did the hammerhead ribozyme evolve once very early, or did it evolve multiple times independently? A single origin would mean that hammerhead ribozymes, is homologous in all those species. Whereas multiple origins would mean that each of those lineages invented the hammerhead structure separately. It's an example of convergent evolution. The question is, which one is it? So many people might intuitively think it's common ancestry. They might think such a complex functional RNA likely evolved only once, just like ribosomes. But what are the chances that random evolutionary processes hit on the same complex RNA structure multiple times? It sounds quite improbable, right? But this has been investigated. Researchers decided to experiment if hammerhead ribozymes could evolve independently, then maybe we can catch them in the action, right? And so that's exactly what we saw in this experiment. This experiment is taken from 2000 and 2001, and this picture is taken from Professor Paul Nelson's presentation. What is the experiment doing, right? So what they do is they bring identical, hammerhead ribozymes, and they kind of try to induce kind of moments of independent evolution of those kinds of ribozymes in an attempt to see if they could originate independently from one another. And so if you can see the results, on the left, it shows the hammerhead ribosome secondary structure, and the right, it's an alignment of sequences from two independent experiments, right? Again, highlighting matching nucleotides. It's illustrating that both evolved RNAs converge at the same functional structure. So, what was the actual experiment? The researchers conducted an in vitro evolution study. They took random RNA sequences and selected for those that could form a hammerhead ribosome. They ran two independent experiments, two separate starting pools of random RNA evolving in separate test tubes. Now, neither experiment had any RNA that was ancestral to the other. So they were completely separate trials mimicking independent origins. The results showed that both experiments succeeded in evolving a hammerhead ribozyme, and the final RNA sequence from the two separate experiments turned out to be essentially the same exact structure and sequence. In other words, the exact same solution evolved twice independently in the lab. This is a complex molecule. The alignment, as we can see on the right, shows that the independently evolved ribozymes had identical key regions. So essentially, nature found the same ribozyme structure two times from scratch. To read the quote from the paper from nature.com, it says, our results suggest that the evolutionary process may have been repeatedly channeled in nature, as in the laboratory, towards repeated selection of the simplest solution. So, again, what they're saying here is that given the constraints of chemistry and function, evolution tends to channel outcomes towards certain solutions repeatedly. Now we have actual empirical evidence to support this. The hammerhead ribozyme is a relatively simple and efficient solution to a biochemical task. And it appears that whether in a test tube or in different species, evolution kept finding that hammerhead structure. This implies that the evolutionary pathway, they were not truly random shots in the dark. They were chemical or functional constraints that made hammerhead a likely outcome when certain conditions were met. Now, the implication of this is vast, right? The implication is that, contrary to what we might assume, the hammerhead ribozyme's existence within multiple different organisms is very well due to independent origins rather than one ancient origin. The improbability wasn't as prohibitive, if given that our initial intuition was that our origination probability was low. We made the false leap into saying that these ribozymes were all descendants of a common ancestor. We were inferring homology when this was actually not an example of homology, this was an example of homoplasy. Now we have empirical evidence to substantiate the point I was making, which is that the less we know about the origination probabilities, the more confident we get about common ancestry. In future episodes, maybe 3 or 4 episodes from now, what I'm going to be doing is I'm going to be providing the logic behind how homology is dictated as opposed to homoplazy. And we'll be touching up a lot more on this during that episode. But this is a good place to gather thoughts and introduce the issue at hand. What this case teaches us is that we can't assume origination probabilities. It's not as simple as saying, okay, because it seems improbable, therefore it's improbable. The hammerhead example is only one in a dozen of peer-reviewed nature journals that show evolution seems to be a repeated thing. It seems to induce exact same repeated genes and structures multiple times, right? So it's not simple enough to just hand wave two similar things as having low origination probabilities and inferring that these are examples of homology. And just to conclude, I brought this quote up before as well. Francis Crick talked about the frozen code, that the genetic code is frozen. And he said something very interesting about that theory. He says, the theory seems plausible, but as a theory, it suffers from a major defect. It is too accommodating. In a loose sort of way, it can explain anything. A second disadvantage is that the early steps needed to get the system going seems to require rather a lot of chance effect, right? The reason why I find this important is because Sober's kind of paradoxical inference runs the risk that Crick just described. It can become way too easy to just say, well, this trait is so unlikely to evolve twice, so it must have one origin. And if it's really unlikely, maybe it's basically a freak occurrence that happened once. So the take home message, at least in terms of this episode, is that the only way to resolve this kind of paradox is to undergo empirical observation, undergo empirical investigation. And now that this is being done in modern research, we're noticing more and more scenarios where evolution or nature is rehashing old genes, old material, old molecules independently from one another. This is why the tide in evolutionary biology is starting to change. If we have time after the series, maybe I'll go ahead and present some of these findings. In terms of the topic of evolution, this seems to be quite novel. It's new research and new data coming to light that seems to be challenging our preconceived notions.